I'd like to introduce our speaker today. Dr. Jan Nolta is director of the Stem Cell Program at UC Davis School of Medicine, and she directs the Institute for Regenerative Cures, and she also is the scientific director of the large UC Davis Good Manufacturing Practice Facility. In 2013, she was recognized and ranked as one of the global top 50 most influential people in the field of stem cells. So we're very honored to be able to have her speak to us about this. Her team at Davis is fantastic. It consists of over 150 faculty members who are working in regenerative medicine-related research, particularly looking at spectrum of diseases and different injuries. And her specific focus is on gene-modified bone marrow, and I'm sure we'll hear much more about that. Jan's experience, obviously, is quite significant in the research community. She's done a tremendous amount of work with NIH, doing different specific review panels for them. She's also been editor, or is editor, for the journal Stem Cells, and was editor for a seminal book, Genetic Engineering, Mesenchymal Stem Cells, if I got that right. But without further ado, our speaker, please welcome Jan Nolta.
Thank you so much. Thanks. Well, I am delighted to be here, and thank you all for coming to listen. So I work with cells and genes in mice and animal models, and hoping to get all of these different therapies into humans, but I am fascinated by technology, so it's just really a delight to be here and get a little later to hear a little bit about what you do. So we do have a large program. There won't be a quiz on this later, but this is just an overview of the different teams we have. And I like to start out with this because it shows our team-based approach to medicine and to regenerative therapies. So we have these different teams that are composed of basic translational and clinical faculty investigators, and working together to try to help their patients that currently don't have many options. So standard medicine, standard of care, can only go so far, and then the MD has to tell the patient, there's nothing else I can do for you. So we start at the bedside, bring that back to the bench, try to develop some kind of novel therapy, and try to take that back to the bedside. The teams that have the green arrows are currently conducting a human cell therapy or regenerative medicine clinical trial at UC Davis. Those with the red arrows, we're talking to the Food and Drug Administration, which involves piles and piles of paperwork. And those with the rainbow arrows have been approved by the FDA, and we're just getting the trial started. So really a robust and busy program. As I mentioned, we start with these problems in medicine, take them back to the bench, try to figure things out, and then go back to the patients. And at UC Davis, since we have the large veterinary school and veterinary hospital, some of our patients are companion animals, and we're doing co-clinical trials with humans and with animals. Some things we can start first in animals and then take forward into the humans, as I will mention. So we've been lucky enough to have funding from the California Institute for Regenerative Medicine. They fund stem cell projects here in California. Lucky to be back in California. I'm a Californian girl. I went away, trained here first at UC Davis, and then went down to Los Angeles for about 15 years. Got my degree at University of Southern California, and then went to the Midwest, to St. Louis, to Washington University, great medical school, but they weren't doing a lot of stem cell research. So when the Prop 71 for CIRM, for California Institute for Regenerative Medicine, when Prop 71 was passed, UC Davis started trying to get me back home, and I was thrilled to come back and to lead this program. And they have been wonderful in funding our facilities, team training, large numbers of students training in stem cells and regenerative medicine, and a lot of our research. They had originally $3 billion through the Bonds Initiative. They have about $890 in new funding to be given out from this year until 2020. And what I'm going to talk about today is that we as Californians doing stem cell and regenerative medicine research are moving into a lot of new clinical trials. And what is critical is measuring the outcomes. After we give somebody a certain therapy, or a batch of stem cells in a certain part of their body, what really did that do? And right now we have surrogate biomarkers, we have patient reporting, we have surrogate endpoints, but if there's some way to better measure each of these endpoints, it will really accelerate the success of CIRM, of our programs, and improve healthcare quite dramatically. And so that's why I'm here talking to all of you brilliant people that understand the technology side of things. Would really like to work together much more. So our facility was funded by CIRM. This is the front of our building. It's not as grand as this one, but it used to be, it's a historic building. It used to be part of the state fair. And when I came back to UC Davis in 2007, it was a big empty warehouse. And we got this grant from CIRM to start renovating it. We finished it and moved in 2010. And we have now about 200 scientists, students, veterinarians, and MDs working in there together. So really happy to be there. This is the diagram of our program. Inside the building is here in this initial circle. We have a lot of cores that we run. The teams at UC Davis that collaborate with us are in the blue circle. And then our outside collaborators are around the edges, different teams in different countries, high schools where we teach and train the students. And we run the statewide umbilical cord blood collection program and are collecting at several different hospitals. So really, every day when I come in to my job, sitting in the middle of this institute, all of these teams are around, and there's something exciting every single day. And I have to say that each of these teams would love to have better measurable outcomes for their patients and for the therapies that they're developing. This is the building. You come in the front here. Our wonderful Clinical Translational Science Center, funded by the NIH, is to the left. And that's where my colleague, Dr. Jim Kovach, who's with us today, works. And they help us with a number of things, translating from the bench to the bedside. Really a wonderful NIH-funded program. The part in blue is funded by the California Institute for Regenerative Medicine. Our name is the Institute for Regenerative Cures. And then we have industry incubator space down at the end where the companies can come in and work with us. We have a industry partner there, PetNet, who does a lot of imaging. And we have the Discovery Lab, the Shared Translational Lab, where I work with a lot of the teams that are coming in and out and doing the final steps before we get into the clinical trial. A large vivarium, a shower-in vivarium, where you completely gown up, and these are all humanized mice. So mice with no immune system that have human stem cells in some part of their body and a human immune system to test the efficacy of the stem cells. And this is where we do all of the toxicity testing for the Food and Drug Administration. And then we're all hoping to get into this amazing good manufacturing practice facility, which is where we make the stem cells. We expand or isolate or grow or engineer the stem cells that go back to the patients. So we're doing right now 11 ongoing and recently completed stem cell and regenerative medicine trials and have about 20 in the pipeline. And this shows the different things that we're working on in the pipeline. So in our building, we are the incubator for the clinical trials, and then in the GMP facility, we actually make the cells and deliver them in usually in syringes for the MDs over to the hospital where they conduct the clinical trials. We have phase 1, 2, and 3 clinical trials ongoing, either investigator-initiated or industry-sponsored. There are some approved therapies. So bone marrow transplantation was the first one. Of course, we do organ transplant like everybody else. There are products for skin expansion and grafting, cord blood transplant. There's a cell matrix for wound healing and a cell plus matrix for bone repair. And by approved therapies, I mean that they are reimbursed by insurance. So these therapies can actually be prescribed and delivered to the patients right now. And we're trying to get these other stem cell and regenerative therapies into that status where it would become care for our patients. We also have a number of cores, fee-for-service cores, to help industry and academic partners move their trials forward. This is a picture of the Good Manufacturing Practice Facility. It's a clean room facility. This is where we get a sample for instance, of bone marrow from the patient. We can grow out the type of stem cells that we like. We can sort the stem cells. This is a immunomagnetic stem cell sorter. It's one of the largest academic GMP facilities in the United States. We have 6 individual suites for manufacturing, and we do fee-for-service contracting and manufacture cell and gene therapies products for national and international academic and industry partners. So keeping pretty busy over there. We can do large-scale cellular manufacturing. This is a Terumo device. It's a hollow fiber bioreactor. And so we used to have stacks and stacks of plates with cells growing in them with media. And now there's this wonderful machine that grows the cells in a hollow fiber reactor and does the work of about 5 technicians just in this one wonderful machine. So we love technology. We have a range of stem cell sources that we use. So we are the kind of the centralized collection and distribution site. Cells are us. So we get the tissues and isolate and help the other investigators work with the cells. So all of the different cell sources that you see here. And also primary tumor samples to understand the nature of the cancer stem cells or the tumor initiating cells in different tissues that come from the cancer patients. We can grow these as xenografts in the immune deficient mice and then test drugs to understand how to kill the cancer. So just so that everyone's on the page, 2 main flavors of stem cells or types of stem cells. They're the adult stem cells and then the pluripotent cells. So the pluripotent cells are very interesting because they can become any tissue. So this is a colony of about 1,000 embryonic stem cells growing on feeders. These are induced pluripotent stem cells. These have been around for exactly a decade now. Shinya Yamanaka got the Nobel Prize for figuring out how to make these. And so basically you can take a little bit of the fibroblasts, the skin fibroblasts from a patient, or some of their stem cells from the blood, and turn them back in time, opening the chromatin so that they behave almost exactly like the embryonic stem cells. And the beauty of that is that they're matched to the patient, so we can understand the genomics of the patient and the phenotype of the cells and put that together and do disease in a dish. The clinical trials, most of the clinical trials ongoing, use these adult stem cells, and that's just because they came first. So the blood-forming or hematopoietic stem cells and the mesenchymal or supporting cells that are found in the bone marrow and in the fat and other tissues. So the induced pluripotent stem cells, this is really all I'm going to mention about those before I go into the clinical trials that are ongoing right now and the need for technology to measure those trials. But these embryonic cells and induced pluripotent stem cells have begun to have clinical trials to treat patients, and these will be expanding over the next few years and the next decade. So they'll be in close step behind the adult stem cell therapies that I'm going to show you. We have a project with New York with the Milstein National Cord Blood Bank to use HLA homozygous cord blood, and that just means that if it's homozygous, there are histology proteins, the human HLA, human leukocyte antigen proteins that are on the surface of every cell, and that's what you have to match for a bone marrow transplant or a kidney transplant or organ transplant. From these homozygous units, they have the same HLA A, B, and C. They just randomly happen to have HLA A15, B22, C65, whatever it may be. And so that means that from these units, 100 of these units would match 85% of the United States population. And then as we add additional units, we could match more and more. So we're very interested in making induced pluripotent stem cells so that we can have this bank held at New York and can match a large number of people with the tissues that can be made from these. Induced pluripotent stem cells are so special because you can make neurons, you can make liver cells, you can make skin cells, muscle, bone, whatever you need to from these cells. And each cell in the colony has this pluripotency, so they're really amazing. Right now what we're doing is making these colonies. We're characterizing them, doing full genome sequencing, and making sure that they are completely karyotypically stable. And then we can differentiate and study disease in a dish from an individual patient. We have some projects going on to study induced pluripotent stem cell induced neurons from children that have different epilepsies and autism spectrum disorder and to understand the nature of their unique disease. And in the future, hopefully, we can differentiate them and transplant them into a patient. And so our leading example of that in my group at UC Davis is that we're making these human liver organoids. And these are liver buds. So we take one colony of induced pluripotent stem cells, make half of it into hepatocytes and half of it into endothelium, and then mix it together. When you put the cells together, they make these beautiful liver buds or liver organoids. And you can see that the vasculature self-forms through them. That is the green here. And this is done by Ping Zhu, who's a junior faculty member in my group. And when Ping transplants these into the mice, these are human, tiny, tiny livers about the size of your thumbnail. They live for 3 months and continue to secrete the human liver proteins and enzymes. So what we're trying to do now is to aggregate them into a larger organ that could be useful for a larger animal than an immune deficient mouse, or hopefully in the future for humans. The adult stem cell therapies have been around, and we've had a lot of years to polish them. Not me specifically. This was a little before I was born. But they began in 1956. That was the first successful bone marrow transplantation. And now hundreds of thousands of lives have been saved by taking the bone marrow stem cells or the blood-forming cells from one patient to the next. We have been doing viral vector-mediated gene therapy since the late 1980s. In 1987, I started working on a project, very exciting, in Los Angeles with Donald Cohn to gene modify cord blood from bubble babies when they were first born, so to put in the broken gene and then return that to the children. Don has since taken that on to functionally cure 23 children that have ADA deficiency, which is an immune deficiency. And now we have many teams working on gene editing for permanent correction of the blood-forming cells. The gene therapy is just taking a viral vector that's loaded up with your cDNA of interest and randomly integrating it into the genome. And now we're able to go in and actually do the gene editing, the targeting, using TALs or CRISPRs. And many teams working on permanent correction of these blood-forming cells. So there are several options that we can do for these stem cell and gene therapy options. We can add the normal gene copy to restore our functions, and the hematopoietic stem cells are currently best for long-term therapy. In the future, it will be the induced pluripotent stem cells as we get more versatile with the clinical trials from induced pluripotent stem cells. We can do the cell replacement strategies, transplantation of healthy progenitors capable of differentiation into the tissue of interest, like the liver, transplantation of gene-modified patient-derived iPSC-derived cells, gene-modified. So if the patient is lacking a certain gene, we might be able to restore it. For instance, people are working on sickle cell anemia, and then can you make blood from that once you restore the patient's own missing or broken gene. We're doing a lot of work with tissue healing, promoting angiogenesis and neurogenesis. So those mesenchymal stem cells that I mentioned, they don't become other tissues. They don't become neurons or hepatocytes. But they are good at secreting factors that promote healing over inflammation and scarring, and they're very easily gene modified. So they're in the tissue transiently, but they can deliver a lot of things to the damaged tissue. And then for the gene correction, the silencing or gene editing for a dominant mutant allele, such as Huntington's disease that my lab works on. This is a nasty neurodegenerative disorder, very bad for the patients that have it. It's a dominant allele. So we need to shut it down permanently or slice it out. And the MSCs can carry the cargo, the gene editing cargo, into the neural stem cells in situ, hopefully in the brain. We're working on that. Many others are as well. And again, the hematopoietic stem cells for permanent correction of blood diseases. So these MSCs are reparative adult stem cells. Sorry if anybody's eating. This is adipose tissue that you can get from surgery. You can get a lot of mesenchymal stem cells out of the connective stroma in the fat. We also get it from bone marrow, harvested from brave donors through going through the iliac crest and harvesting. We start out with a non-differentiated, non-homogeneous population and have ways to purify them, sort them, grow them out. And I had published a book on their genetic engineering about a decade ago. and still using those same types of modified strategies. So these cells are very cool because they have a paramedic function in the body, and they like to communicate with other cells. So I'll show this video, watching this area here. They do a lot of cell-to-cell contact. So these two just joined up through tunneling nanotubules. And if you can see closely, there are actually mitochondria going between the cells. They can transfer not only what's in their cytoplasm, but whole organelles from cell to cell. They leave behind these things called microparticles and also exosomes. And we have a team led by Jonathan Anderson in my group that's working on exosomes. These are microparticles. This cell will take up what that cell left behind. They get extra ATP and mitochondria, and then they divide, and then they go on to communicate with other cells. So we're capitalizing upon this nature of these cells to deliver whatever is inside them, whatever's on their membrane, whatever is in their cytoplasm. They can deliver it pretty robustly in a local area where we inject them into a damaged tissue. And their function is to try to keep the other cells alive and promote vascularization. And we have a lot of evidence in vitro that they cause neurite outgrowth from neural stem cells as well. And so all of these things that I just mentioned, another fascinating thing that they do is that they keep the immune system at bay during tissue remodeling. They reduce inflammation. So it's thought that, we have these throughout all of our different tissues and organs in our body, and it's thought that they really do have a paramedic function. When there's an injury, they're first on the scene. They migrate pretty quickly, as you saw. That's a picture taken every 30 seconds. They're first on the scene. They really keep the immune system away from that area so that you don't get autoimmunity against things that are there in the injury, denatured collagen or whatever they might get excited about. But really, these cells are not becoming the damaged tissue in most cases, but they're acting through their paracrine effects and everything else that they do to the other cells. We've had some statistically significant successes in some human clinical trials, and then others have been not significant in the field and make a lot of headlines when they're not significant. And so we're trying to figure out how to best grow these cells, how best to deploy them into the damaged tissue, and especially the outcomes. Once we do that, how do we exactly, precisely measure that there's better angiogenesis, that the injury is better? And so that's what I'm really talking about today. We get a lot of cells from adipose tissue. There are stem cell clinics out there that are just taking adipose, like liposuction from the patient, and just kind of cleaning it up and re-injecting it. Not, I want to choose my words carefully, not usually sanctioned by the FDA. You should go through the FDA and show them that you're doing things in a very sterile manner. You're doing it in the same 24 hours. If you're doing that, not adding anything to it. So really the way that the cells are being handled, and if you do more than minimal manipulation, they have to go into one of these good manufacturing practice facilities before being returned to the patient. So this was a very nice review in Stem Cells Translational Medicine by Dr. Laboa and her group. Now, we have worked for a long time, as I'd mentioned, with the Veterinary Institute for Regenerative Cures. And the Center for Equine Medicine has a great regenerative medicine program. They have a sports injury team. They work on tendon, ligament, cartilage, soft tissues, and bone. Dr. Larry Gallupo is the head of this Center for Equine Health. We've worked with him for a decade. They work a lot on the bone repair team. This is Kent Leach, who's in our School of Engineering, and he makes these different matrices that you can add the cells back into bone to have it heal much more quickly, funded by the U.S. Army and the NIH. They've piloted all of these novel matrices and cell therapies in the horses, horses that have a hairline fracture or a bone cyst. Mark Lee is our physician over on the human side of the hospital. He's working on the research as well and performing stem cell trials of matrix and bone repair in human trauma patients. And so you can take the human bone marrow and isolate it into different fractions, add it with some different matrix, and it causes the bone to heal much more quickly and more durably. This is a little hard to see because there's so much on it, but this is our sports medicine team. This is a really exciting team. Jim is the leader of this team, having been a NFL player in his past before he decided to go and get his law degree and his MD. Great, great combination. I think you're probably the only person I know that has all of that background. But this is a great team because we have been working on sports injuries in the horses at UC Davis for over a decade now. We do the therapy, the horse's own stem cells, and this is a very future planning, hopeful slide because we hope that some of you can help us find the best wearable technology for these horses and also for our human athletes to definitively know that the stem cells that we're giving them are making them better. And right now, we have human athletes. They are getting, when they get an injury, they're getting their platelet-rich plasma, which has a lot of those little microparticles and vesicles and all the good growth factors in the plasma. We want to soon start a clinical trial using mesenchymal stem cells for patellar tendon chronic injury. A lot of people have that, and we need to measure outcomes to know if the athlete is ready to go back to play, and also just anybody that has this injury and we're treating, are they doing better? And so we really need definitive measurements. Right now, the key outcome that they're using, they use ultrasound to look at the tissue. That's not that specific. But they have a pain scale that's self-reporting. So this is inadequate and it's not definitive. And you basically ask the patient, what is your pain on the scale of 1 to 10? So we really need to look at maybe symmetry. Are they favoring one leg? Shoe insoles I've seen, looking at the speed of movement, ability to climb stairs, any of these things where the patient could be monitored throughout the day with wearables to give the MDs definitive continual data to know if that patient started doing better after the stem cells were administered. So we do the next generation MSC products, and what are these? MSCs that have been culture expanded and then genetically engineered to enhance their secretome, what they're putting out, or their function, or MSCs implanted on a matrix or other carrier for combination products. So this is a picture of mesenchymal stem cells in Integra, which is a matrix for wound healing. We are making biological bandages. This is a program funded by CIRM, and these are for patients with diabetic non-healing ulcers. We do a lot of gene editing, gene therapy in these cells because they can be easily engineered to secrete high amounts of protein and other factors for delivery to target cells and tissues in the body, including the brain. We are working on loading them up with cargo and delivering it into the brain, for instance. We are working on a study of intramuscular injection of cells, MSCs, making vascular endothelial growth factor for the treatment of critical limb ischemia. And this is a CIRM-funded project with my co-PI, Dr. John Laird, who runs our vascular center at UC Davis. And this is what happens when there is not enough blood flow down into the leg. This is common in diabetes and can happen to older people who don't have the proper circulation. We have a mouse model of this. We use the human stem cells to try to restore the blood flow down into the limb. We have an animal model where we can actually cut off the blood flow down into one leg. We use laser Doppler imaging right now, so you can see the blood flow, and we measure at specific times. But if there are better ways out there to measure blood flow in mice and then in people, we would sure love to know about it and maybe test some of those technologies. So we can show that when we use these gene-modified MSCs, they do better than the controls at restoring blood flow. And then when we make them in our GMP facility, again, the red is the gene-modified MSCs, significantly restoring blood flow. We've done a lot of what are called farm tox studies for the FDA, using the GMP-grade vectors and MSCs, and these are all the different things that the Food and Drug Administration wants to see from us. We had what's called a pre-IND, pre-investigational new drug meeting with the FDA in 2013, and then Had a meeting with the, submitted all of the data to the Recombinant Advisory Committee, and then had the pre-IND in 2015, hopefully moving into clinical trials in 2017. So these things take time. We have time to figure these technologies out and the best measurements for the outcomes for the patients. And so this is our team that's working on that. Fernando Fierro is the lead on that, and Julie's our surgeon. We have some great industry partners, and of course, CIRM's been funding that one. But again, we need these better measurements for definitive and earlier outcomes. Right now, the outcomes for the planned trial are if the patient has wounds, as I showed you, are they healing? And the main outcome is that we don't amputate the patient's leg. And that's a pretty broad stroke. You know, it would be nice to have some finer detail and finer measurement. Looking at better ways for oxygen perfusion, finer tuning. Again, the wearables, looking at stride length or walk duration might be good outcomes. I had seen that the blood pressure wearable devices might not be that accurate, but better ways to measure the blood flow in limbs, things like this would really help for these clinical trials that are ongoing and being planned. And then the last one that I wanted to mention was using the mesenchymal stem cells to deliver a neural growth factor, a brain-derived neurotrophic factor, as a potential treatment for Huntington's disease. In Huntington's disease patients, this is a very nasty neurodegenerative disease. It really takes its toll on the patients and on families. And the level of this factor, brain-derived neurotrophic factor, plummets to very low levels in Huntington's patients because their mutant protein actually acts back on the manufacturing of the protein. And BDNF is needed to keep the neurons alive, to keep them connected, to keep neural stem cells turning over, and it's a pretty important factor. And so we've been hoping to engineered cells to produce this, to put them into the brain of the patients, and we have had a CIRM-funded project currently ongoing with the pre-cell that we're doing right now. And we've done a number of CIRM-funded studies. We can show that in immune-suppressed Huntington's disease mice, the human MSC BDNF reduces the striatal atrophy that happens to the mice. It increases neurogenesis. There's a behavioral modification. They have reduction in anxiety, for instance, and extends the lifespan. So we are moving forward. The Food and Drug Administration has asked us to test the delivery using the exact catheter that we will be planning to use in the patients on 3 pigs. And so we are currently working on that. Just have published this paper in Molecular Therapy showing the outcomes of testing our human cell product in the transgenic mice that had to be immune suppressed to allow the human cells to engraft in them. And we've had this cohort of amazing Huntington's patients ongoing and measuring them for about 1.5 years now so that we can see if anything changes after they get the stem cells. And so they are going through MRI. They are having spinal taps to look at the CSF fluid to see if we can find biomarkers that might go up. We're doing testing. They're doing brain imaging. And they come in once every 6 months to have all these tests done. If there was some way to understand if they're doing better through technology, this would really help make the trial much more definitive. So right now we're doing a volumetric imaging analysis, and we can see over time these blue areas are degenerating in the patients. And we do this with Charles DiCarli's lab at UC Davis, which leads all of our imaging. And here again in the yellow, you can see changes in brain volumes just over 6 months. These striatal areas are really diminishing. And so we need things, first of all, we need some therapy to stop that or prevent it or delay it, and then we need really good ways to measure it. And so we are trying to move forward with this. We had our pre-IND meeting with the Food and Drug Administration, and we're doing these additional biosafety and dose finding studies right now to go forward. We have a gene editing team for juvenile Huntington's disease. This is led by Kyle Fink, who's a really awesome postdoctoral fellow in my group. And they can go in, here's their paper that just came out, they can go into the patient's fibroblasts and reduce just the mutant allele through using TALs, or now through using CRISPR-Cas now. And the challenge at this point is to get this gene editing cargo into the brain to affect the neural stem cells that are in the brain, and then again to measure outcomes after that. So what we need, these patients have chorea, they have different jerky movements, they have balance issues, indecisiveness and anxiety, they have problems with spatial awareness, gait analysis. This is also very important in Parkinson's disease, and maybe there could be future tech interventions as well as just measuring what the stem cells do. For instance, there's a bionic spinal cord brain technology interface that was just reported. So things like that seem so exciting to me, but I don't really understand it. So yeah, thank you. So awesome, great. Is it, you know, how the biological signal can be converted into electricity? That's fascinating to me. So I look forward to talking about that. And we have a Dr. Ki Kim, who's fabulous, who's doing actually three stem cell clinical trials now. Another one just opened, and is really excited to test new things. He's great. So in this new era of medicine, prescriptions will be and are already being written for stem cells to treat these intractable diseases and injuries. And there are expanded cryopreserved bags of these MSCs approved to be prescribed as a drug in Canada, New Zealand, and Korea. And reimbursement starts during the phase 3 trial in Japan now, and this is through Mesoblast, who assumed all of the technology from Osiris. And improved outcomes measures will really accelerate all of our progress. So this is our team. This is everyone in the institute, in the program that works in all of the cores and works in our shared translational lab and vivarium and GMP facility. We meet every Monday morning to organize what we're going to do that week. And just a lot of eager, young, enthusiastic, and wonderful people. And that's all I have, so thank you. Any questions, or we need to get out of here pretty soon?
Wonderful.
Yeah.
We'd like to, I think we have mics here. We'd like to open it up to questions from the audience. Thank you so much. That was not only impressive, but opened some very intriguing opportunities. So for those of you, we have a mic. We'd love to entertain questions for the next few minutes. So please, the floor is open. Want to repeat
you can yell, I can repeat it, yeah.
Thank you to excuse my bad English. I'm a French surgeon.
Great.
Very interested in your topics and in your results in angiogenesis with stem cells. Do you have clinical trials with results in human trials?
Do we have results yet? We
Yes.
we haven't started the trial with the genetically engineered product that I was introducing, the MSC VEGF, but our vascular center is doing two, and soon to be starting on a third stem cell clinical trial with industry partners, and so using different types of stem cells. And so they do have all of the infrastructure in place to measure the outcomes that we have, the oxygen perfusion and the wound healing, and but very interested in other technologies that would be faster, more definitive. We need numbers for these things.
And just one more question: do you use it in diabetic patients?
A lot of the patients are diabetic, and that's why they have the reduced blood flow.
Great.
Yeah.
Thank you.
Thanks. Look forward to talking to you more about it.
my name is Elise. I'm a Dutch biomedical student.
Hi.
And I just finished a research proposal on how these pluripotent stem cells, the induced ones, can help functioning for treatment resistance in depressed patients. And one of my major questions for my proposal was, are they really going to reflect the treatment resistance and the treatment response to antidepressants? So have you found any results already that the cells really reflect the patient?
Yeah, that is a great question, and of course, using these for mental health issues is the next big field. And so you're right at the right time for that. So far, I only know of definitive testing for things related to epilepsy and neural disorders where the signaling is not quite right. So, for instance, from Fragile X tremor-associated syndrome, we had a paper where we took iPSCs from the patients, made neurons, and then showed that they had a more sustained, when you did the patch clamping, they had a more sustained signal. They didn't do the spike like they should. But the response to different hormones, with just the individual lawns of neurons, I'm not sure how far that goes. People are now making brain organoids that might better reflect because they have the cells interacting and connecting to one another. Wildly fascinating area, but I don't have the answers in that. It's, we can't get the neurons from the patients to try, so this is the best we have. And, congratulations for working in that. That's really interesting.
Hi, I'm Stephen McKinley. I'm a roboticist, and I was wondering, are you using systemic or localized injections of these stem cells for your therapy? And if you're using localized, how important is the positional accuracy of those injections?
Yes, great question. We use both depending on what the indication is. For intravenous injection, though, a lot of the MSCs, which are large cells, they'll get filtered out in the lungs. So if you're not trying to treat lung disease, you really should put them into the area. For our angiogenesis study, we'll be doing a grid of 29 different injections intramuscular into the leg, and the positional accuracy is really important. And maybe, Jim, you want to say a few words about for the sports injuries? We were just talking about that this morning. They use ultrasound to see where the injury specifically is.
In the initial clinical indication is going to be a patellar tendinosis, so like runner's knee. And you can, with today's ultrasound, you can actually identify very specifically where to make the injection. That is important. It needs to be tested, but if you think of the other injuries that we're tracking beyond that would be rotator cuff would be a very prevalent one. Tendinosis of the ulnar, ligaments in the arm, but yeah, currently the state of the ultrasound has gotten good enough to where you can basically put the needle in and inject.
Yeah. And then I have to say, for the brain, of course, that is exquisitely important. You need to get them exactly into the right area. For Huntington's, it's the striatal areas.
Yes. Hi.
Hi.
In your initial long list of disease treatment areas, you mentioned the lungs. Could you expand on the pulmonary picture? Thanks.
Yeah, pulmonary is one of the areas that, at least with the mesenchymal stem cells, from the IV injections, they can have the best results in the lungs. So for COPD and other trials, the stem cells, mesenchymal stem cells, were being delivered intravenously to try to treat a heart attack initially. And it was found that the best result that they had was to help with the lung function for the patients. And that's because those of us who had done the tracking of the MSCs knew, but I guess in the patients we weren't quite sure that about 90% of them will lodge out in the lung during the first pass. They're pretty big cells. They get stuck in the capillaries. And so there's a lot of work being done there. We have a lung team. Reen Wu runs a lung team and is working on development of lung tissues from the embryonic stem cells at UC Davis. And then there's some amazing work being done here at UCSF on lung disease. nearby at UCSF.
Any additional?
Anything else?
Well, we would like to thank you very much for a very stimulating presentation one more time.